introduce It's my pleasure to introduce Adriana Brown, who is a junior in our department, majoring in environmental geoscience. And so she's been working with us, started over the summer and is continuing somewhat this semester, working on our NSF-funded research project studying intensively managed agricultural landscapes. So she's going to tell you more about the project and about her work on it.
Thanks, Dr. Welt. So as she said, I'm Adriana and I'm from Purdue. And today I'm going to be talking about the critical zone, especially looking at tile drains and water chemistry and what we've been doing with the critical interface network. So the critical interface network is a multi-institutional research collaboration that really looks at the critical zone, which is considered the part of the environment that's from bedrock to the tops of trees. So you can see on this diagram here includes rock, water, soil, organisms, air, everything in that critical zone on the environment. And then specifically the critical interface network, SciNet, looks at water, carbon, sediments, and nutrients and how those are transported in the environment. Another focus is that SciNet looks at different interfaces within the critical zone. And these interfaces are areas where different processes meet. So they can be chemistry, biology, geology, but what they all have in common is that it's an intersection of multiple scientific processes. So an example is in the active root zone, which is the root zone of trees where they're taking up water and nutrients into the plant, or a floodplain, where when rivers and streams flood, they move out into the soil, and they can be natural or man-made. An example of a man-made interface is the tile drain network, which we'll talk about more in this presentation because that is what we focus on in this project. And one thing that SciNet talks about is that even though these interfaces aren't necessarily large in area, they exert a very large influence over the entire landscape. And so they have a disproportionately large influence over our environment. So studying the critical zone interfaces gives insight into dead zones that we see in our oceans. And as you may know, the dead zone is simply an area in the ocean with too little oxygen in the water to support life. You can see on this map here that there are coastal dead zones as well as open ocean dead zones. And as we study these because they are a result of some practices in the Midwest. And a little bit more on the dead zone process is that it originates from fertilizer nutrients draining into waterways and ultimately the ocean. So to walk through the steps of that real quick, when there's a crop that needs fertilizer, that fertilizer often contains nitrogen and phosphorus, which are the two important chemicals when we're talking about dead zones. And as that crop, the water becomes saturated and it doesn't absorb any more of the water, the runoff water carries those fertilizer nutrients and it then leads into waterways and streams. And then streams carry it to the oceans. And then when it gets to the ocean, those fertilizer nutrients cause aquatic organisms to grow really fast. And when they do that, they use up all the oxygen in that ocean area. And that's when you see like the algae blooms. If you've heard of the red tides in California, there's an example of one here in this image. And so that's kind of the large scale effect of this specific process that we're looking at. So to bring it closer to home, when we think about the Midwest, we think of agriculture. And the Midwestern agriculture heavily contributes to the dead zone in the Gulf of Mexico. Indiana alone contributes about 10 to 15% of the nitrogen in the Gulf of Mexico. And so when we think about this, there's two main goals of land management, just overarching goals. And one is to reduce the application of nitrogen and phosphorus fertilizers because those are the relevant nutrients. And then also to reduce the transport of nitrogen and phosphorus off the fields and not get them into the rivers and streams. So one way that agricultural land is managed is through tile drains. And tile drains, have an image here on the left, are ways to shortcut natural water flow pathways. They keep the field from flooding, and they're used really commonly, especially in the Midwest and Indiana. And as you can see from this diagram here, it's pretty much bringing water directly that drains from the field into this tile drain and out, discharges to a stream. So you're not getting as much of the subsurface interaction as a natural water process might have. Indiana, when we talk about northern Indiana, about 50 to 75% of it is drained by tiles. And you can see looking at this map that centered in the Midwest, it is heavily tile drained, which makes thinking about nitrogen transport really important, especially when we know that there's a dead zone in the Gulf of Mexico. So what we do know is that tile drains increase the nitrate entering waterways. You can see from this graph here that as we move throughout the months of the year and we look at tile drained area, that the more area that's drained by tile drains, the higher the nitrate will be. So it's the top plot here up on the line. And then when you think about it overall, that the more tile drained agricultural land, more nitrate concentrations. And so again, that's kind of what we study, because eventually they do end up in our oceans and create dead zones. And so thinking back about the agricultural fields, water can take different pathways through the soil to get to the tile drains. This is an example here of macro pore flow versus matrix flow. And macro pore flow is when water moves really quickly through large cracks in the soil. It is, like I said, a faster process, the cracks are larger, and there's not as much chemical interaction with the soil particles. On the other hand, matrix flow is when water moves a lot slowly through smaller cracks and crevices. It lingers and it can chemically interact with the different soil particles around it. So what we've been doing in our project is focusing on the problem that excess runoff and nutrients in excess runoff contribute to poor water quality downstream. And this can come from agricultural runoff, urban runoff, atmospheric processes. But the ultimate question is, how does water flow pathways through the soil and the tile relate to chemical composition? So what happens between the soil or the water running off the field and the tile drain, and what chemistry is happening during that process? So the way we do that is we look at tile discharge hydrographs, which is looking at the volume of water moving through the tile over time. And we do that using water stable isotopes, electrical conductivity, as well as nitrate concentrations. So we use water samples from Allerton, Illinois. The Allerton tile drain is there, and we study the critical zone in an agricultural field. So before we began this project, they've been previously analyzed for different chemicals like nitrate and different anions and cations. And then now we have access to continuous measurements from tile drain discharge, volume, and electrical conductivity. And when we talk about electrical conductivity in soils, that's really a measure of salinity and how many dissolved ions are in it, like nitrate, salts, chloride, and in a given area. And then that determines how well the soil will conduct an electrical current. You can see on this diagram that our water is sampled from the structure for water control. And that's where we get like our discharge estimates to see how much water is moving through that area and where our samples come from that we've studied the chemistry of. So focusing on what we've done in our project so far is think a lot about prior conditions of the soil, because that can really alter the pathways that the water takes through the soil. Just an example of an event from the summer of 2016, August 15th. And here we'll see evidence of water moving through macropores and then transitioning to water moving through the matrix. So kind of looking at what we have here, in the top left is a plot of nitrate concentration over time. The right is electrical conductivity. The bottom left is oxygen 18, which is a water stable isotope. And then the bottom right is tile discharge. And these four graphs together kind of tell a story about this event, that we see a sharp increase in discharge when it was raining. This event in total got about a ½ inch of rain. And as that rain increased and more water was moving through the tile drain, the electrical conductivity decreased here. And that's because the soil and the water was diluting the different compounds in the soil. And then we look at nitrate, and we see that it starts a little bit after when the highest volume of water was moving through the tile is not the same time as the highest concentration of nitrate. There's a bit of a delay between peak discharge and peak nitrate, and we think that's because of macropore flow, which basically the water was moving quickly through these large cracks. It wasn't picking up nitrate as it went. It was more of a fast movement where it wasn't chemically interacting as much. And that's why we don't see nitrate peaking with discharge. A different event happened in the spring of 2017 on March 30th, and it's received about an inch of rain. And when we think about spring, the soil is typically a lot wetter. There's a lot fewer of large cracks and more smaller pathways that water can take. And that's why, one of the reasons why we think this event had matrix flow. So we have the same plots here as before. And this time when we look at nitrate and discharge, they increase at the same time. So as the volume of water was increasing, the nitrate concentration also increases. And we think this is because as the water was moving through the cracks, it was able to mobilize nitrate as it went, because it wasn't just rushing through large dry cracks, it's more falling through crevices. And that's why the trends in those two variables are similar. So thinking about where we're at now, like I mentioned earlier, we are still focusing on the problem of water quality caused by excess nutrients in runoff. And some of the future work is that although we focused a lot on specific precipitation events, like I showed in the last two slides, kind of what happens in between those events and what kind of biological and microbial processes are happening. So we've been looking specifically at different events, looking at different chemical tracers, such as nitrogen-15 and water-stable isotopes, to try and figure out what exactly is happening on the microbial aspect of agricultural fields in the critical zone. So with that, that's all I have today, but I'm happy to answer any questions if we have them.
Thank you, Adriana. Are there any questions from the audience for Adriana?
I do.
Okay.
For the runoff, is that typically farmland, or is that like no-till?
Adriana,
Do
did
have
you?
one versus regular farming as opposed to no-till? Would there be less because of the cover crops and stuff like that as far as runoff?
Adriana, did you hear that question?
Can you repeat it for me, please?
Yeah, the question was about no-till versus traditional agriculture and whether or not you see a difference in the runoff between the two. Is that correct?
Yeah.
Yes, okay.
Yeah, I believe the field that we're on, Dr. Wilkin should probably know more about this than I do. But is it till no-till that we're looking at?
Yeah, I'm gonna have to look this up. I know we have it's a corn soy rotation. So I'd have to look her 2016 and 2017 data. It might be a different crop. I don't think they're doing cover crops at this particular site, but those are really interesting comparisons to do if you have sort of paired sites set up with different land management. Those are really great experiments. At this particular site, we only have one land management to study. So there's an additional part of the project that's looking at a prairie where we don't have the tile discharge, but we can kind of peek into the soil and see some of the chemistry that's going on in the soils and the prairies and compare that to the ag soils. It's a great question. It does make a difference.
Do you have any other?
I can tell you one way that it makes a difference. I'm just people are curious. So it actually turns out in the no-till, you get more of these big cracks that develop. And so with the tillage, you get fewer big cracks. So it kind of, it changes the water flow through the soils. There's lots of reasons to do no-till, but it has different effects on the water transport.
Thank you. Are there any other questions from the audience? Oh, I see one in the chat from Logan, and it says, is there any way to mitigate the large chemical concentrations before it reaches the Gulf of Mexico, or do we have to catch it at the source?
I think from what we've identified that one of the more efficient ways to do it is just managing runoff and nutrient transport at the agricultural field site. But I'm sure there are people working on mitigation processes farther down the line.
I'll just add to that and plug some of the work that I think NGRREC is doing, right, in studying wetlands. So adding in wetlands in the landscape can kind of be this intermediate catch for nutrients. So yeah, we can do things to reduce the non-point sources a little bit everywhere, and then hopefully add in some kind of catch points as well. That's the goal.
Thank you. Any other questions from the audience or over Zoom? Right, we have one more.
Go away in the future, like a wastewater treatment plant that we use for our drinking water on a large scale. When this gets real bad, 20, 30 years down the road, somewhere down there in Mississippi. Is there any new chemicals you can add to Mississippi that would dissolve some of the bad stuff or make it better or anything? Do fertilizer companies research any of that kind of stuff to mitigate damage that's being done, different kinds of fertilizers?
Yeah. So the question is whether or not there's a system such as a water treatment facility that could filter out some of these nitrates and things like that before they make it down to the Gulf of Mexico. And then whether or not there's any like chemicals being developed that would help dissipate those, the nitrogen or phosphorus or break it down. And whether or not there's any agricultural companies looking into whether or not those things can be mitigated. Did I get your whole question?
Yeah, that's about it.
Okay. Yeah, he said 20 or 30 years from now, is this something that would be feasible?
They may have to do something.
All right.
Honestly, I'm not super knowledgeable on if there's any sort of water treatment facilities going on.
I don't think so.
A lot of water. Logistically, that's quite the big ask. So I'm not an expert in ag management, but I think the goal is to do kind of the right timing of the fertilizer applications to make sure as much of it gets into the plant and less of it is available to move out into the waterways. So the timing and the type, and the more we like, nitrogen is very, like it changes forms, like with microbes, it changes forms really quickly. And so making sure that we understand how the microbes are changing nitrogen and what's available to the plant and when, I think is going to help in some of those processes about applying the right thing at the right time.
Thank you. We have another question from the audience here. The question is what type of fertilizers are being applied to the crops, whether it's just manure or if it's something else.
That's something because I haven't had Adriana study all of the history of the site. These are synthetic nitrogen fertilizers. So anhydrous ammonia, I'm forgetting, there's like 2 different ones that they can apply. Some of it's applied in the fall, some of it's applied in the spring. I'd have to look up the records to see. And it's different if it's corn versus soybean, they apply different things. So, but yeah, so at this particular site, it's not manure.
We got another question.
Has there always been a dead zone? Or when did it start? Or is it just getting bigger?
So the question was, has there always been a dead zone? When did it start? And is it just always getting bigger?
We definitely have seen them increase with agricultural practices because we are sending more nutrients into the streams and waterways. And I think it is a change that we can see in our lifetime. So we have seen them change and expand over like a decade scale.
These are great questions because I'm gonna have to look up like when it started. It's been going on for a while. The interesting thing that I do know is like wetter years mean bigger dead zone. So there's the bigger volume of water that you get moving out the Mississippi, the bigger the dead zone is that year.
You guys ready for another question?
I'm gonna write down all my like questions for next time. That's awesome.
Well, we're recording, so you can rewatch it too. All right.
It's another question, just a comment on the dead zone and the nutrient stuff. There wasn't always a dead zone because before the river would spread out into the Delta of Louisiana. And it's just dammed up all the way, or levied up all the way out, so it shoots out straight out to the Gulf. And part of that, kind of sky treatment of nutrient pollution and sediment is actually a big diversion project that's going on in Louisiana to put big diversion systems in the levees instead of it keeping on out, that actually spreads back out into the wetlands, the swamps that it used to do help create land in Louisiana with sea level rise and
something like that.
Did you guys hear that over Zoom?
No.
Okay. It was a comment from one of our audience members who actually used to live down near the Delta. And he was saying that prior to as much channelization in the river as we have now, there was not really a dead zone because the water was being dissipated more through different wetlands and things like that. But now, since we have levees and other things built up across around the river, it just gets channeled right down into the Gulf without going through any of those natural systems. And if you look at the Mississippi River Delta, due to the sediment loss, it's been shrinking. So the water doesn't get spread out in more of a shallow area to mitigate some of the effects that contribute to the dead zone. Did I summarize it well? Okay. And it looks like we have
Those are really awesome points. Yeah, your giant wetland is your big water treatment facility. Yeah.
So our next question is, how long does it take the dead zone to recover, or does it? Is any project in place to educate the farmers?
I think we see them fluctuate based on like times of year. They'll be stronger at certain points in the year. I don't know if it ever fully retreats, but there definitely is points where it's at a maximum or a minimum.
Yeah, there's that natural seasonal variability. I'm under the impression that if nutrient discharge was to be reduced, that the dead zone would respond really quickly. There's not like a super, within the marine environment, there's not a super long legacy. However, on land, there is a long legacy. So this is something that we talk or that we study, not me personally, but the field. So if you reduce fertilizer application, you can still have this like stored nitrate within the soil, nitrogen within the soil, or even the aquifers. And so even if you like shut off the fertilizer each year, it would take a while for the legacy nutrients to kind of wash out of the soils. So there's the lag in that bit. But if you were able to reduce it, I think the marine response is pretty quick. There's a lot of education that goes on with farmers. I mean, I know my grandparents were farmers, and no one is like putting extra fertilizer on for no good reason, right? I mean, it costs money. So there's a desire on all sides to reduce usage as much as possible because it helps the bottom line. We're just getting like more and more data-driven ag to understand exactly what's needed and where it's needed. So there are definitely efforts. Yeah, this is not an unknown problem.
All right, we have one more question. So this is the last question.
I see a lot that you're talking about on the agricultural side of the runoff, but on your project, you're talking about the urban runoff and the atmospheric. How much of that is participating in this, or is it all agricultural?
That's a great question. This is actually my fault because this is kind of a generic slide that I've used for other projects, other things. So in this particular one, urban runoff, we can eliminate. There's a little bit of atmospheric deposition, and so there's an add-on to the project. We'll be collecting rainfall to quantify that, but it's really pretty small compared to the direct application that's done on this site. If you go to like a natural forest that no one is applying fertilizer, atmospheric deposition is a bigger source then. So my apologies for the misleading wording on the slide.
Well, thank you very much, Dr. Welp, and thank you so much, Adriana. For those of you that might not know, Adriana has been interning with us at NGRREC and the CINET project for about a year now. She started at the beginning of the summer last year, so she has really followed this research quite a ways, and it's really nice to see that an undergraduate intern is so passionate about this topic. And I will say one more thing, if you want to learn more about the dead zone in the Gulf of Mexico, you can check out the NGRREC website of Great Lakes to Gulf as part of our Great Rivers Ecological Observatory Network. And we have some data on our website of Great Lakes to Gulf pertaining more to the dead zone and runoff and how it changes the dead zone. I believe that data goes until 2017. We don't upload our new data right away because we like to hang on to it. All right. Well, thank you guys so much.
Thank you.
All right. So we have our next intern. Her name is Betsy Papen, and she has been working with Dr. Danielle Hake, who is the director of Illinois River Watch. And she has been, well, I'll let her tell you what she's been working on, but she's from Godfrey, Illinois, and she is a criminal science major at Lewis and Clark Community College. And she comes to us fresh off an internship with the Alton Police Department. So we're lucky to get her here and we appreciate her work. All right. Let me just get this set up for you. All right, can everybody see that? Can people on Zoom see that? Yeah, okay. All right, There you go.
Thank you. Set this down here for now. All right, so hello everyone. I am Betsy Papen, like she mentioned. My internship project has been on Prairie du Pont watershed. Just to give a little bit of background information, Heartland's Conservancy is in the early stages of a 2-year watershed planning and best management practice implementation for the Prairie du Pont watershed and neighboring Judy's watershed. These watersheds are located in the northwest corner of the St. Clair County, including the communities of East St. Louis, Allerton, Washington Park, Centerville, Westview, and Dupo. So western areas of this watershed are located in the American Bottoms, the floodplain of the Mississippi River. Allerton, Centerville, and Cahokia have joined to form one municipality. So I will be using their names kind of interchangeably because that's pretty recent. So what exactly is the problem? So over 30 years, infrastructure in the areas of the American Bottoms have been failing, resulting in mixing of stormwater and sewage, including the ejection of sewage onto residential streets, basements, and lawns during storms. Community organizations like the Centerville Citizens for Change has been working to raise awareness and initiate the processes needed to take care of the flooding and sewage spills. So this slide kind of just gives a few different news articles that shows how important and how insane some of this is. So like horrendously inhumane, Cahokia Heights resident files new lawsuit in effort to fix chronic flooding and sewage problems. That's just an example of one of the headlines. If you Google this, there's quite a few you can get some information off of. So just to give you a little background, an article by St. Louis Today highlights 74-year-old McArthur Smith pictured here. He is a Centerville resident and for years has dealt with this sewage problem. Even the slightest rainfall results in him spending hours scooping water with gallon buckets inside of his house. His wife has a disability that makes her immobile, and when the flooding occurs, the toilet is unable to flush, and his wife has to use a bucket that he takes to public restrooms to get rid of. So these are just a couple more photos that show the extent of the flooding right on people's houses, streets, and lawns. So in support of the watershed planning efforts, Heartland's Conservancy has teamed up with NGRREC and Illinois River Watch. So the goals of this partnership are to connect with residents with their local water resources, empower residents with skills needed to measure local water quality, but especially this is the last one is the one I focus mainly on my internship is provide baseline information on the current condition of streams in the watersheds. So when providing that baseline information, my job is to test the watershed locations for fecal coliform in specific. So fecal coliform density is the standard for measuring in the state of Illinois when testing for bacteria. There is a normal amount of fecal coliform found in natural waterways, but if there is a high trace of fecal coliform, there is a chance of other harmful bacteria that are not as easily to test. But testing for fecal coliform indicates the possibility of those more harmful bacteria in the water, especially human sewage. So these are the two watersheds. North is Judy's Branch watershed, and underneath is Prairie du Pont watershed. So these are some of the sampling sites. There's 7 sampling sites. We have multiple to show. Some of them are creeks, and we also have lakes, creeks, kind of just to show differences. Then we have testing. So when testing for the bacteria, a sterile water sample is collected, and not only is fecal coliform being tested, but we're also collecting information on the pH, conductivity, water temperature, dissolved oxygen, using different types of equipment. So everything is documented and put into a spreadsheet. This is just us me out at different locations, taking water samples. I liked my overalls because they helped carry everything easier. Once all the samples are collected at each site, they are taken back to the lab here and receive a packet of chemicals that lets us know if it's yellow, it indicates that there is fecal coliform present. So go to the next. Once the packet or the sample is given that packet of chemicals, it is put into a tray, sealed through a machine, and then put in an incubator. So 18 hours later, the next day, the results are ready. So any of the wells that are yellow show a positive count, while any that are less than yellow are negative. So all of the large wells and the small wells are added together to give you the amount of fecal coliform in that watershed. So there is a grid we use, and I also brought a sample just to show, this is one I collected yesterday, just to show what it looks like. So on the back is the location and the time that it was taken. If it's positive, I put a slash through it, and then all of these are added. So let's say there was 48 down vertically, I go to 48, and then if there's 3 small wells, I go horizontally and match the numbers on the grid to get the fecal coliform count. So on the left here is just to show what a great result would look like. So those have no less than 1 amount of fecal coliform present. The middle is kind of an indicator on if it's that yellow or more, it is positive for fecal coliform. And on the right was results from one specific location that had a pretty high amount that week of fecal coliform in their water. So these are the results as of last week, the 16th. So down horizontally, I can't really see you too well because of the right here. It has the dates down here. So the dates, as well as vertically here, has the amount of fecal coliform in the water. Up here, we also have the bar graph that measures the amount of rainfall around that date. So if one specific date spikes, we might be able to see the rain also coincides. So some dates, it does show that higher rainfall resulted in a higher fecal coliform count, but some days, like last week, the numbers were all fairly low, even though we had a high amount of rain. So it just kind of depends. So in Illinois, the fecal coliform count is 200. So up here you can see we did surpass that number quite a few times, but looking back, there are multiple dates where the way that we test is not the exact same way that the state of Illinois tests. They test those results multiple times and they find an average or a mean. Ours is just kind of a baseline of information to give them an idea of what we're looking at or what we could be looking at in certain locations. So it's not done the exact same way as the state of Illinois tests. So like previously mentioned, the location of the sampling, this was not the locations that we originally had in mind. There were some locations that we could not have because of restrictions. So we found some other locations that we thought would be great. So these did not have the highest amount of fecal coliform count possible that we were expecting, but because of where we picked, some of the restrictions that might have had higher amounts, we didn't get to test at. So this kind of gives us, it's nice at the same time because it lets us know that these locations we can exclude from whatever issues they could be having in the area. We know that these locations might not be having that problem, or it's not coming from here. But what exactly is next? What does all of this mean? So all of this information is going to go back to Heartland's, and they are creating a watershed plan development on what to do with this overall huge problem. So they're creating this watershed plan development and then working with the citizens and community to decide where these grant and funds go, where they want to prioritize it, which areas need it the most. And then they work together to put that grant money where it needs to go. So we're also hoping that this internship and this experience opens up the gateway to, in the future, opening volunteer bacteria monitoring. So different areas and different community members, whoever is interested, would be able to take these samples in their local waterways and understand or get an idea of what they're looking at near them, what water they are around. And we're also hoping in the future that some of those locations we may not have been able to get to, or that could have higher numbers, we may be able to get to in the future and can get a better understanding and a better idea of where these things are coming from or where they're leading to, these problem areas. So for my acknowledgement, I'd love to give a shout out to Danielle Hake. She's not present today, but she, as well as Jennifer Riemann from Heartlands Conservancy and Danielle from National Great Rivers. I'd love to give them a shout because they assisted me in this project, as well as multiple staff members from Heartlands Conservancy and National Great Rivers. So this is just some references and some links for different news articles that talk about this that I've used throughout this presentation. So if anyone wanted to check it out, you can also just Google it. There's quite a few articles that reference the situation. Any questions? Yeah. So it's kind of what we're looking at is if it's lower numbers, then there is supposed to be a normal amount of feces in natural waterways. We have geese. We did, one day we saw that there was multiple geese feces all over the water. So there is a normal amount in natural waterways because of animals and things like that. But if we're seeing a high amount multiple times, or we're seeing especially high amounts, it's most likely human sewage that's in that waterway. Yeah. Oh, I'm sorry. Yeah. So that was regarding if it's possible human sewage or animals, or what exactly type of sewage or feces we're discussing. Yeah.
My question is, what kind of restrictions?
It's just about like ownership and who has rights over what lands, and there is some legal issues going on between the municipality suing the different. It's a lot of legality, and so to ask for permission, it's kind of like private land. If they say no, it is what it is. It's a good question. That's kind of why we wanted to sample it. So the question was, why did?
But that was most of it is basically, we just don't want you.
Yeah, it was, yeah. So the question was, why would they restrict us from certain areas from testing? And some of the areas I know were government, some of them were private property. It just depends. But yeah, it was just, they had the decision. I know that they had some legal issues, or there was EPA is involved. And so everyone's trying to figure it out. I think they wanted some toes in it, and I'm not.
And that all runs right into the Mississippi, I guess, everything you're checking, I guess essentially has to go there.
I'm not quite sure. I know there are some lakes that are just lakes. I'm not sure where everything runs. There was a couple of creeks that there was one down in Dupo called Fish Lake, but it's actually a creek. It's a little misleading. And then there's Schoenberg Creek. I'm not exactly sure where they lead to, but that's kind of the, what we were looking for was, okay, if this location is here, where is it coming from and where is it going to? Because if it does, does have a high amount and it is leading to the Mississippi or this area, then that's something we need to pay attention to.
And what do they,
No, you're okay.
what do the favors causing all of the flooding? The fact that there's just water, the fact that the storm drains and the sewer drains.
Right.
Run and I'm throwing heavy rain, it can't handle all the water.
So the question was, why is it flooding to the extent that it is? And I think it's kind of a perfect storm of a couple things. Like I mentioned, it is in the American bottoms. So it is naturally a very flat, low area that has had problems, over time flooding. But I know there have been issues in the past for over 50 years from my research of some of those things failing in the area, like pumps and things that should help. It's just so overworked because of the flooding that a lot of those have failed. And so when it does flood, that sewage is getting mixed with that flooding that might not be as harmful if it wasn't mixing with some of the things that are outdated and have problems. I think that's part of this project. I think that's part of the goal. Yes,
I
ma'am.
believe if it wasn't for pumps, keeps all that water, I mean, we would not be living in those areas, even in Granite City. If it wasn't for the pumps pumping all that water out, all the way the whole area that you're at too.
Yeah.
You just wouldn't get there without pumps pumping that water out.
And I could totally be wrong because my job was kind of just focusing on the water sampling in these areas, but yes, but from what I've learned, there are some problems with these pumps because of how old they were and then how significant the flooding is, that it cannot take on the amount of water that it's flooding. So it's hard to keep up and it's not juggling the right way. So that's something they're looking to fix with this grant money, is doing all of this research so that with that grant money, they can figure out which pumps they can allocate the right amount to and which areas need more money. Yes,
You
ma'am.
don't want to track how many human, like E. coli from this, like, because people don't wash, they clean up and then they may not wash their hands or anything. They don't track that, do they? Like, how many if humans get sick from being in that, with E. coli. or something.
So the question was if anyone is tracking
Yeah, that.
the E. coli and stuff like that.
Humans are, like that poor guy that's cleaning up after his wife.
Yeah.
Well, they do, but do they actually track it? Yeah, well, I know, but do they, I want to say track.
I'm not sure about like certain areas where it's some of these neighborhoods and stuff like that where this flooding is more, if they're keeping up with the health conditions and who is in charge of that. And if I know that Centerville Citizens for Change is documenting quite a bit, and I know Heartlands is documenting quite a bit of what's happening and then the effects of what's happening. So I'm sure that they're keeping an eye on it, but I know that the state of Illinois is supposed to monitor these waters as well and has a standard that they're supposed to meet. So I'm not sure if those, because they're neighborhoods and not technically waterways, if that's, I might not be the best one to answer that, but that's just my understanding.
All right, last question, and it comes from Zoom.
Okay.
What locations are you expecting to have high levels consistently, or are there any locations?
So there were a couple locations that had higher numbers, but not exactly the most insane that we were expecting. Anything urban is kind of where we're focusing, where there's more houses, more neighborhoods, schools. Anything urban is kind of where we're expecting to see, because more humans equals more human sewage. So we're expecting in those areas but some of those areas where there were neighborhoods and stuff like that, we were not able to get to. So we're thinking that those areas that would have the higher numbers, we couldn't exactly test from. So we're hoping that in the future, that's something that would be able to look into.
Well, thanks that you're doing that stuff. When people like it.
All right. Well, thank you. Thank you, everybody.
Thank you so much, Betsy. And we have one presentation left, and I'm going to have Dr. Justin Chu introduce his advisee. And let me just pull up his slideshow. All right, here's Dr. Chu.
Thank you, Sarah. Well, my name is Justin Chu. I'm the Conservation Program Manager for the National Great Rivers Research and Education Center. So you've heard talks a lot about water quality today. So I manage teams actually that work with some of the agricultural producers throughout Illinois that put some of these conservation practices on the ground that can definitely improve water quality. I like it because those same practices can provide wildlife habitat. So we're going to segue into a wildlife talk tonight, and I'm pleased to introduce Jacob Futrell. Is that right? From Staunton, Illinois. He is a freshman at Lewis and Clark Community College. He is in the general engineering program, a pathways program leading to eventually going to the University of Illinois. I learned that Jacob learned about this internship, and a relative showed him this internship in the paper. So thank you, relative, if you're out there, for introducing me to Jacob. I learned that Jacob and I have a few things in common. We both have played soccer. We both love to backpack, so that's cool. I get to talk to him about those things. But I'm gonna let Jacob take it away, and he's gonna tell us about what he's been working on for the last semester.
All right. Yeah, thank you. My name is Jacob Futrell, and my project was over studying eastern whippoorwills and their habitat. And since I'm going for engineering, I kind of did it on more of a technical scale. So I looked at them through using acoustic monitors to keep track of them. So the acoustic monitors would be used to determine whether they were at a certain site that we had them stationed at. But I guess the first thing we should look at is, what exactly are eastern whippoorwills? Well, an eastern whippoorwill is a small insectivorous bird, meaning that they eat insects, mostly moths, and they're part of the nightjar family. They live in this area. They are from Eastern North America and they're migratory. So they migrate south to Mexico in the winter. And they are also nocturnal, and more specifically, they're crepuscular, which is kind of a new word. It means they are mostly active around dawn and dusk. So the twilight hours is when you're mainly going to see this bird. And I actually have some audio right here, and this is what their call would sound like. So that's just kind of what their call would sound like. And that's also an example of what we'd be listening for on these acoustic monitors. And that's a picture, a nice picture of a whippoorwill right there. It's during the daytime, so it looks like it's resting because they're nocturnal, but just a little bit more about them. They also, interestingly enough, they nest on the ground. As you can see, there's a picture of one of their nests right there. It's kind of exposed out in the open. There's little materials used in it, but they do nest in the ground, and there's a picture of their hatchlings as well. And additionally, kind of an interesting fact is the fuller the moon phase is, the more active these birds become. So if you see a lot of the moon phase showing, these birds are probably going to be more active because they kind of like twilight, just a little bit of light, and the moon provides that. So if there's a full moon, these birds are probably going to be out because they like the extra light. Now on to the preferred habitats. Like I mentioned, they're native to Eastern North America, and this map shows that. So the red area is where their breeding area is, and that's where they'd be during the summer. So we are well within that area, and so it's possible we could see some around here. And that map also shows they migrate south to Mexico during the winter because they are migratory. And more specifically, they do like open understory forests, specifically on like forest edges. So like the larger the forest patch is, like along the edges of the forest, you'll find a lot of whippoorwills. That's their like preferred habitat. And they also really do not like any human interference. So any areas that have a lot of development, any urban areas, even croplands, like if there's fields nearby, they don't tend to live in those areas. Probably because light pollution really messes with their sense of timing, because they have a very narrow window to hunt. They only hunt at dawn and dusk, so any light pollution could mess with that. And also their prey is very sensitive. So any insecticides or herbicides that are being sprayed near it may drive off their prey. So they're very sensitive to any human interference. So why do we need to research whippoorwills anyways? Like, what's the purpose behind this? Well, a whippoorwill, along with a lot of other types of birds, including many birds in the same family as them, are endangered. Well, I shouldn't say they're endangered. They're declining. They're a species in decline. And like this graph shows here, this red area is all places where they are declining by 1.5%, over 1.5% a year, which is a pretty steady decrease in population. So we do need to keep in mind that these birds are decreasing. And to make it worse, we also don't know a whole lot about these birds compared to other species. They're like relatively unknown due to their odd hours of activity. They're harder to research. So we don't exactly know what the causes are of their decline, but we do have some ideas. The main culprits we think would be habitat destruction and prey loss. This picture here is of an owlet moth. It's native to the area and is probably a prey of the whippoorwills. And due to what I mentioned before, herbicides and pesticides being sprayed, the moths and other insects are being kind of driven out of their areas, and the whippoorwills, they don't have as much prey to hunt, so it's kind of hard for them to keep population up. And as well as this graph right here shows surface area use in Illinois. So as you can see, the vast majority of it is cropland, and only about 11% is forested, which is the whippoorwill's habitat. So these birds don't really have much space to work with overall to begin with. And even though this is just a graph of Illinois' land use, most Midwestern states and Eastern states, which is where they live, have the same statistics. So it's not an isolated occurrence. Onto our study. We, like I mentioned, are using the acoustic monitors to keep track of their presence. These monitors will be active during twilight, dawn, like twilight hours like dawn, dusk, and then midnight. We're gonna have them set up so they record 10 minutes around dawn, 10 minutes around dusk, and 10 minutes around midnight to get as many recordings of them as possible while not having to listen to like our whole night's worth of recordings, because we have to listen to all this ourselves. So however long the recording goes for is how long we're going to have to listen to it. So the goal is to try to maximize the amount of calls we might hear while minimize the amount of time we have to spend listening to these recordings. But these pictures right here are examples of sites that we have used. The one on the left is a site at Lewis and Clark Community College. And this is actually going to be a test site we're going to use in our actual study. Unfortunately, we have not been able to catch any whippoorwills yet on audio because there's typically they come around end of April. So far we've mostly been troubleshooting and testing these sites out, but this is a site here that we will be having this monitor at. The monitors themselves they're produced by Wildlife Acoustics. It's a manufacturer, and the specific model is the Song Meter Micro. They're about $250 per unit, and we have 4 units of them. You can see the front view on the left. In the middle, there's the interior of it with the lithium batteries. And on the right, we have a side view with a microphone, which is how the recordings will actually be captured. These monitors, they allow for a lot of variation. So I think specifically, I would focus a lot on trying to like maximize the amount of whippoorwill calls we can hear by like changing the time settings on them. And I'm able to make the monitors specifically record whenever the sun goes up and down, because these monitors are actually smart enough to know what exactly the daylight schedule is like whenever the sunrise and sunset are like at our like area. So in Illinois, it can detect when the sun rises and sets, which is pretty interesting. Also, due to the like nature of the study, unfortunately, we won't be able to test the number of whippoorwills at each site just because we can't actually see any visual cues or anything. We can only hear, and it's kind of hard to determine the number of birds at each site. So we will only be testing for habitation, not the number of them. Also, kind of as a side note, any data that is taken during rainy or windy conditions is going to have to be thrown out because the sound quality might be damaged too much by like the loud rain and wind. So the area of study itself, we're going to be using 9 sites, 6 of them at Pure Marquette State Park, which is to the left. That's just north of us. And then 3 sites at Lewis and Clark Community College campus, which is the picture on the right here. And these sites, the most important thing is they're sites that we already have a lot of data on. They've been used for many studies before this, which is important because we have a lot of data on the microhabitats and the land on a smaller scale. So we have like data like ground cover or any invasive species in the area, which is important because a lot of studies on whippoorwills, especially, don't include this kind of data. They use a lot broader visuals like satellite visuals. They can't determine all these habitats on a smaller scale. So with these sites that we have a lot of data on, we are trying to get a better idea of what these birds like on the smaller scale. And like I mentioned, invasive species are present at some of these sites. Specifically, bush honeysuckle is a major problem in this area. It's an invasive species and it's very prolific. It's spread a lot. And we don't really know the effect of invasive species like this one on these birds. And since they roost on the ground, it's possible that species like this one can crowd them out, interrupt their nesting habits. So we really want to conduct this study to determine whether species like these will have a negative effect on these birds. And this also ties in well with a lot of NGRREC's other activities. For example, the NGRREC strike team would go out and clear bush honeysuckle like this at a lot of different sites on state and private land. So this could also be something that is a long-term project for NGRREC overall. We actually do have a few recordings gathered by the acoustic monitors. Unfortunately, there's no whippoorwills as they're not in season yet, but I do have a recording right here with some, I guess the audio file does not want to play. It says it no longer exists. So you're gonna have to take my word for it, but that is the audio file for this recording here. So like this sonogram here is of that audio file. And you would hear bird chirps at the higher marks. So wherever the red tick marks are that like dot up, that is where the birds would be like making more noise or any other background noise. We could, yes. If we listen to the recordings, we could. The question was whether we could identify what birds these are. So yeah, we could do that when we listen through them. We don't know yet. The question was what bird is it? We don't know yet what bird it is. Yeah. But this is our study, it does have several differences from other studies. So for example, this study right here was done near Ottawa, Canada, and it tested the habitation of eastern whippoorwills and nighthawks, which are another bird very similar to whippoorwills. And these like sites are indicated by the smaller dots is where eastern whippoorwills were present, and the larger dots are where nighthawks were present, and the X's were where neither bird was present. So as you can see, this study, like it spans a very broad range. It has a lot of different types of land there. It has cropland, forests, shrublands, a lot of different variety. And this study, it covers a way larger range than we are going to in this study. But what we lack in quantity of sites, we make up for in quality, because this study in particular used satellite visuals, which are a great way to get an overview of the land at a macro scale, but it did not reveal a lot of data on the undergrowth or a lot of the smaller things like ground cover. So our study mainly focuses on getting a better view on the smaller scale and determining this, what might affect the whippoorwills coming to these sites on a micro habitat scale, which really hasn't been done much before. So this is kind of a novel study. We're trying to determine this, and with all the extensive background research we have on these sites, we should be able to get a good picture of the habits of these birds. And this study, the acoustic monitors in particular, it doesn't have to just be used for this study. There are also several other applications after the study is completed for these monitors. For example, there's gray tree frogs and barn owls in this area as well, also have very audible calls. So these monitors could be used to test habitation of these, even bats or other birds that are endangered in the area. We could use these monitors for basically anything that makes a noise, honestly. So there's a lot of possibilities for this, and this is only the beginning, really. We're just testing whether this method works, and if it does, hopefully we can apply it to more species and make a bigger impact. And finally, our goals for this study are mainly to gather data on the habitats of these whippoorwills, what exactly they're looking for in a habitat, confirm the presence in the area, because it's kind of hard to do a study on whippoorwills if they don't live here, and overall to learn how to better protect them. These goals are fairly vague, but we hope to continue this study on and gather information about them so we can form a better plan of action as we go. For acknowledgements, I'd like to thank NGRREC for the funding for this, Dr. Justin Shu for helping me basically through every step of the process, Melissa Schindler, Jennifer Hemphill, the NGRREC strike team, Sarah Fisher, and Amy Monroe. And if anyone has any questions, I'd be happy to answer. Thanks. Yep. Yeah, so the question was, does the bird have whiskers? It does have whiskers. Yeah, it has whiskers, yeah. Yeah, so they kind of as a side note, they do hunt, like they're aerial insectivores, so they catch these like bugs out of the air. So they need to be very precise when they hunt, and they likely use the whiskers for that, because catching a bug out of the air probably isn't very easy. Yeah, they are nocturnal, so it makes it even harder at night to hunt for these insects.
So Jake, when you're hearing this on the audio, and you hear them chirping or whatever, are they like mating calls, or what are they actually doing while they're chirping?
Right. So the question was, when the whippoorwills are making this audio, this activity, what is the goal? Are they trying to make mating calls or what? So the answer is a good part of it is mating calls. Specifically, whenever it's mating calls, it's called churring. It's a term for it. It's kind of a niche term, I guess. But yeah, a lot of it is mating calls potentially, and a lot of it is just them being birds and calling anyways. But yeah, it kind of all plays a part, right? And we're going to be listening to any of that. So, yeah.
Like territory defense.
Mhm.
Is that a male or female, or is there a big difference? You know, usually in the bird species, the males are more glamorous. Is there a difference, do you know, in male and female?
Yeah,
You would
so.
try to see them, you know, sometimes.
So the question was whether this bird right now is that's being shown as a male or female. And the answer is, I don't really know. I think that the males and females are fairly similar. I never encountered anything that said otherwise. So I'm assuming that they're pretty similar in terms of like appearance. Yeah.
And they have that like a white under their tail there, it looks like.
Yes. Yeah. There is traces of white underneath their tails. So if you see that from below, then there's a possibility it's a whippoorwill. Yeah.
Are you monitoring, you said you were doing it more on a micro than a macro. Are you monitoring the insects and the bugs for food sources at the same time that you're listening to the birds?
Yeah, so the question was, during our study on a micro scale, are we going to look at the insects in the area and like their possible prey sources, right? So we really don't have a way to look at the insects in particular because this study mainly focuses on using audio, the recorders, and we would really have to like go in person, like do some like on the ground work, go there at twilight to find whether these insects are present or not. So we really can't get that good of a look at their prey at these sites, which is a good point. It might skew our data, but I think one of the main goals is to try to test these audio recorders out to see if they actually work and are like viable as a source of like, keeping track of these birds. So.
Are there any questions in the chat? There are not. Well, thank you so much.
All right.